So my name is Sriram. I'm a faculty at UCLA. And the reason I'm giving this talk is because my student Ali, who led this work, couldn't make it to the workshop. So what I'm going to talk about today is a technique called variance components analysis. It's a widely used technique in human genetics. And the reason why we started thinking about these kinds of methods is in the context of the fact that we have what we call biobank scale data in human genetics. So what that means is today we have data sets like the UK Biobank, which measure genotypes from hundreds of thousands of individuals. We have thousands of traits or phenotypes that are measured. And the challenge is how do you design interesting statistical models to take account of all of these interesting data sets, and then the algorithmic and inferential issues. And so that's what led us to think about variance components analyses. So here's an example of the kind of problem that we are trying to answer. So this is something called heritability in human genetics. So the concept of heritability is trying to ask how much of a trait or phenotype comes from genetics versus the environment. So we're trying to decompose a person's trait into some function of the genotype plus the environment. Of course, this concept is much more general. You can think about heritability in the context of other measurements, other features, for example, microbiome or gene expression. And the best way to think of this is it's just a quantification of the signal inherent in your feature set. So this is an abstract concept, and the way we actually think about it is we ask, what is the best linear predictor of your phenotype given genetic data? So concretely, we have phenotypes that are measured on a bunch of individuals, and we have their genotype. So this is typically SNP data, so it's a matrix. And we are trying to understand what is a linear function of genotype that explains the phenotype. So each of these SNPs is associated with an effect size, and everything else goes into the environmental component. Now the problem, of course, is this is a high-dimensional problem. Typically, we have millions of SNPs, thousands of individuals that we're dealing with. So to be able to handle this problem, we put a prior on the effect sizes of the SNPs. So a typical prior is every SNP is assumed to come from a Gaussian distribution with some zero mean and some variance. So this variance is what we call the genetic variance component. The environment, again, is assumed to come from a Gaussian distribution with zero mean and a variance, which is the environmental variance component. So having defined the genetic and the environmental variance component, we can estimate heritability. So heritability is one number that you compute given a data set of genotypes and phenotypes. Of course, you don't have to restrict yourself to this one number, and there's been a lot of work in trying to understand how heritability is distributed across different parts of the genome. For example, there's been work trying to figure out which chromosomes contribute to heritability, how heritability is distributed along the genome in different genomic elements, and, for example, whether it is shared across different traits. So the way to think of all of this is we can extend our variance components model. So instead of the effect size of every SNP coming from a single Gaussian distribution, we might add different effect sizes depending on additional covariates associated with these SNPs. For example, we might say that if a SNP is in a coding region, it has one distribution. If it falls outside, it has a different distribution. So in generality, we have a variance components model which looks like this. We have the phenotype, and we have a collection of SNPs which are assigned to k different variance components. All the SNPs in a given variance component share variance parameters, and then we have the environmental variance component. So given the setup, our inferential goal is to estimate the k genetic variance components and the environmental variance component. How do you do this? So this is a well-defined probabilistic model, so we can write down the marginal likelihood of this model and optimize it. So this is what is done in typical methods today. And in fact, the likelihood itself tends to be biased, so there are modifications of this called the restricted likelihood that people typically optimize. So the challenge, of course, is this process is computationally intensive, so you have to do a numerical optimization where each iteration scales cubically in your sample size. So for the kinds of samples that we care about, where n is greater than hundreds of thousands, these kinds of methods tend to be infeasible. So we decided to use an alternative estimator. So instead of looking at maximizing the likelihood, we look at the moments and try to match movements. So this is what we call a method of movements estimator. So this is not something new. This has been proposed in classical human genetics. It goes under the name of Haseman-Elston regression. So the idea of method of moments is simple. You have the theoretical moments of your model, and you choose parameters so that theoretical moments match your sample moments. Turns out the way we have set up the model, the first moment, the mean is 0, so we look at the second moment. And the second moment can be written as a linear combination of the variance parameters weighted by these matrices. So there's one matrix for each variance component. It's an n by n matrix. And this is what we call the genetic relationship matrix. So it's an n by n matrix where every entry tells you what is the inner product between every pair of individuals at the subset of SNPs that you're looking at. So now we choose our variance components to match the sample moment. The sample moment here is obtained from the phenotype. So what's the advantage of this approach? Turns out the method of moments estimator is available in closed form. So we can actually write down a system of linear equations that gives us the method of moments estimates of the variance components. And what do we need to compute the coefficients of the system? It turns out they have this form. So the coefficients are typically looking at products between our genetic relationship matrices, and we are computing certain functions of these products. So it seems like we are done. We compute a method of moments estimator, and we have our answer available in closed form. Not quite. It still requires computing these n by n genetic relationship matrices. So if you have m SNPs and n individuals, this leads to a quadratic time algorithm, which is still not feasible for these data sets. So the key insight that we had was we'd like to avoid ever computing this genetic relationship matrix. And the reason we can potentially do so is the sufficient statistics, the only quantities that you need to compute these method of moments are not the GRMs, but the traces of the GRMs. So all we need to be able to do is to be able to estimate these traces efficiently. And there is classical work showing us how to estimate traces, going back to Hutchinson's estimator. So these are randomized unbiased estimators for traces of matrices. So we can take our original trace and approximate it by these randomized estimators. And the key computation is we need to look at our original genotype matrix and multiply it by a small number of random vectors. So now we have an additional parameter for this algorithm, the number of random vectors that we need to be able to estimate these traces. So there's going to be a trade-off between statistical accuracy and efficiency, depending on the value of B. And what we find is for the kinds of data sets that we are looking at, we get very accurate results for B as small as 10. So to sum, we have an estimator of heritability or variance components, which is essentially linear in the size of our data matrix. So now let me talk about some results. So we started out by looking at the accuracy of this estimator. So we looked at small scale simulations where we can compare our estimator to the gold standard. The gold standard here is maximum likelihood implemented in this package, GCTA. And we looked at different kinds of what we call genetic architectures. So different distributions of effect sizes, different heritability parameters. And what we find is both the randomized Haseman-Elston regression that we propose and GCTA are unbiased. Our estimator has slightly larger standard errors than maximum likelihood, which is expected. The reason for this is it's method of moments, so it's not using all of the information that's available in the distribution. However, the key thing is the maximum likelihood does not scale to larger data sets, so then we turn to large-scale simulations. So now we decided to assess the accuracy of our method on large scale, so we are looking at about 300,000 individuals. At these large sample sizes, the only methods that work are methods which compute different kinds of summaries of the data. So these are methods that summarize the data and operate on these summary statistics. And again, we looked at different genetic architectures, and what we found is across these architectures, the randomized Haseman-Elston regression is unbiased, whereas all of these other summary statistic methods have fairly large biases. And we can talk about the reasons behind this, but primarily this is due to the approximations that we're making when we're computing these summary statistics. So then we looked at the scalability of our estimator. So this is the runtime as a function of sample size compared to methods which are maximum likelihood based. And what we find is even for 300,000 individuals, which is the typical sample size that's available in the UK Biobank, our method runs in an order of hours. So looking at it a couple of different ways. So we explored the runtime of the method for a given sample size, which is available in the UK Biobank, about 300,000 individuals, 500,000 SNPs, when we are fitting about 300 variance components. So that's the other major factor. We also explored what happens when you dial up both the number of individuals and the number of SNPs to 1 million. And again, all of these computations can be done on a typical desktop. So then we turned to some real data analysis. So we looked at 22 complex traits that are available in the UK Biobank. And we estimated genome-wide heritability from our method, and again compared to these summary statistic methods. We find that across these 22 complex traits that we analyzed, methods are more or less concordant, but we do find that our method tends to yield lower heritability estimates on average compared to these other methods. So one of the things that we can do here is not just estimate heritability across the genome, we can also understand which parts of the genome are contributing more or less heritability. So here we did this analysis, which is quite interesting, where we divided up the genome according to what we call the LD patterns around these SNPs, as well as their frequency. So we have SNPs that are low frequency versus common, and we have SNPs that have low levels of LD, so they're not very correlated to nearby SNPs, to those which have high levels of LD. And we can ask how much heritability comes from each of these bins. And what we find is common SNPs on average explain more heritability than rare SNPs. On the other hand, as the level of LD decreases, there is an increase in heritability. So again, we are not the first to document this, but this has been seen in previous works. But what we see is an extremely strong signal because we are able to analyze data at the scale of the UK Biobank. And there's different kinds of hypotheses about what drives the signal. The primary one is this is a signal of negative selection acting on the traits that we are looking at. So just to summarize, what I've talked about is a scalable way of estimating variance components. In practice, we find that we can estimate hundreds of these variance components on biobank scale data in a few hours. And the ability to fit multiple variance components allows us to fit expressive models. We don't have to limit ourselves to simple models. And as an example, we fit models which show how heritability varies across different minor allele frequency and LD architectures. Turns out this kind of approach to variance component estimation is actually quite general. It's useful in lots of settings where variance components are used. So my student Ariel had a poster here on multi-trait analyses where this can be used. And this can also be used to estimate things like gene-gene interactions, association testing, and others. Finally, of course, there's interesting technical connections. So these models, you can view them fundamentally as Gaussian processes with a specific kernel. And so there's interesting connections on how this can lead to scalable Gaussian process inference. All right, with this, I'd like to thank all of the people involved in the work, but primarily Ali, my PhD student, and Ariel, who's here and presented a poster on extending this to the multi-trait setting. Thank you, and I can take questions.